afternoon, fellow researchers. My name is Natnicha Sunantawanich, a graduate student from Department of Biology, Faculty of Science, Chiang Mai University, Chiang Mai, Thailand. Today, I would like to talk about efficacy of LAC products and bioactive compounds on anti-inflammatory activity. First of all, I would like to talk about LAC products. They are extracts from LAC, the resonance compound secreted by LAC insect, Laccifer lacacer. This insect normally lives on host trees such as rain tree, longan, lychee trees, and etc. It distributes in India and Southeast Asian countries. In Thailand, we can find it in North and Northeast regions. LAC product has been used in many forms. For example, lacca has been used for Ayurvedic medicine. Shellac has been used for surface coating, tablet and capsules. And lac dye has been used for food and fabric dyeing and also for cosmetics. For lac extraction, stick lac would be discarded the waste or the stick. Then to get lacca, lacca will become lac dye, the red pigment, and seed lac, the resinous part. After that, seed lac will be further extracted with 3 different processes. First, decoction to get wax containing shellac. Second, ethanol extraction to get dewaxed shellac. And third, bleaching extraction to get white shellac. In our study, we are interested in 3 LAC products, which are lac dye, seed lac, and and dewaxed shellac. Even though LAC products are useful, they are rarely studied specific on LAC insect morphology and life cycle, constituent compounds, compound isolation and properties, and biological properties such as antimicrobe and anti-cancer. For example, this study, the researcher study on the structural identification and their bioactive activities. They found that 2 members of raising group and 3 members of lac dye group are active on antibacterial activity, but inactive on antifungal activity, and yet no report on nitric oxide inhibition and so far no study on anti-inflammation. When we talk about anti-inflammation, we need to talk about inflammatory cascade, which started with inflammatory stimuli such as lipopolysaccharides or LPS. The stimuli trigger the inflammatory signals through an F-kappa-B pathway to express inflammatory gene expression, especially COX-2 and INOS, which are the critical enzyme for inflammation. With arachidonic acid pathway, COX-2 would produce PGE2, and at the same time, INOS would produce nitric oxide. These 2 substances cause the inflammatory symptoms, such as swelling, fever, and pain. If we can inhibit the inflammatory gene expression, we can inhibit the inflammation. The objective of this study is to investigate anti-inflammatory activity of LAC products and their bioactive components. For the method and results, I would like to start with the sample preparation, then cytotoxicity test by using MTT assay, then inflammatory gene expression using qRT-PCR, and at the end, nitrite analysis using Grease reaction. For sample preparation, LAC extracts, lac dye, seed lac, and dewaxed shellac from 2 sources, rain tree and lychee tree, would be dissolved with dimethyl sulfoxide, DMSO, into 40 milligrams per milliliter, and standard compounds, lactic acid and allylic acid, would also be dissolved by the MSO into 40 millimolar. This solution will be stored at 4 degrees Celsius, then further diluted with serum-free medium into this series of concentrations. And MTT reagent would be dissolved with phosphate buffer solution or PBS into 1 milligrams per milliliter and tightly sealed. These solutions will be freshly prepared. For cytotoxicity test, we use MTT assay. The law 264.7 milli macrophage cell line would be plated in 96 well plate and treated with standards and samples. After 24 hours, MTT reagent was applied, and after 4 hours, the absorbance was measured at 570 nanometer by microplate liter. At the end, the percent cell viability would be calculated from this formula. According to ISO 10993 part 5, greater than or equal to 70% is non-toxic. And the result shows that in the lactide group, only 40 micromolar of lactic acid is toxic. And at the same time, in resin group, only 20 and 40 microgram per milliliter of rentidivastelac are toxic. Therefore, we chose up to 10 microgram per milliliter and up to 10 micromolar of samples and standards for the further studies. For inflammatory gene expression, we use quantitative reverse transcription polymerase chain reaction or qRT-PCR. The cell was plated into 6-well plate and pretreated with lipopolysaccharides to trigger the inflammation, then treated with standards and samples after 1 hour. After 24 hours, the culture media were collected and the RNA was extracted, then converted into cDNA and measured the gene expression using qRT-PCR machine specific on COX-2, INOS, and GAPDH genes. From the lactide group, we can see that all treatments except 5 micromolar of lactic acid could potentially suppress COX-2 gene expressions and also INOS gene expressions. On the other hand, in resin group, we found that COX-2 gene expression could be suppressed by allylic acid, rentidivastelac, and 10 microgram per milliliter of rentidivastelac. And for INOS gene expression, all treatment could potentially suppress. To confirm the INOS gene expression result, we analyzed the nitric oxide production using Grease reaction. The safe culture media from qRT-PCR method was mixed with Grease reagent, then incubated for 10 minutes and measured for the absorbance at 550 nanometer. And as we can see that all treatments could effectively reduce nitric oxide production. So we found that black products and bioactive components could potentially reduce inflammatory gene expression. Lactines are more efficient on cell viability and COX-2 gene expression, with lactine as the most efficient. Therefore, lactine is good for anti-inflammation. At the same time, for resins, they are more efficient on INOS gene expressions and nitric oxide reduction, with rentidivastelac SLR as the most efficient. And moreover, allylic acid is more effective on anti-inflammation than lactic acid. This might be the effect of the different proportion of the components in like resin, such as teloic acid, lactolic acid, lactolic acid, and etc. Therefore, like resin is good for antioxidant. Moreover, we found that the sources of like products are matter, supported by the study of Kurana in 1970 that shows the different proportion of allulonic acid from different sources. In conclusion, our study showed the efficacy of like products and their bioactive compounds on anti-inflammation, which could promote the pharmaceutical purposes. Finally, I would like to thank Smart Insect, International College of Digital Innovation, Department of Biology, Faculty of Science, Chiang Mai University, and my advisor, Dr. Hatachanok Pandit from the same department for the support. Thank you for your attention. Feel free to ask if you have any questions.
Thank you for your wonderful presentation.